So welcome to this video 7 in our introduction to transcriptomics. So with this, we're going to briefly talk about the practical that we're doing. So this practical is based on a study from last year, 2 years ago, on the induction of antiviral and cell-mediated immune responses significantly reduce viral load in acute foot and mouth disease virus infection in cattle. So this is a particular bioproject ID, and we're going to get the data from there. We can go to that bioproject ID and download those files from the SRA by getting their file extension names that we can then use the SRA toolkit via FastQ dump to retrieve those FastQ files. And what we're dealing with there is 15 samples in total, all of which are taken from the ventral soft palate tissue of the cow, where foot and mouth disease virus levels can be measured. 3 breeds are examined with 5 samples from each of the 3 breeds, hence 15. 2 of these are Bos indicus, the Malad Gida and Alicar breeds. And then the other breed, those 5 cows are Holstein Friesian and they're Bos taurus. So we need to consider the foot and mouth disease infection status as well as the breed in our model. And I guess the question is the immune response higher in these calves, leading to a milder FMD serotype? And we should note as well that this serotype does not necessarily mean that they will have a different immune response or change in immune response between these different breeds to other foot and mouth disease serotypes because of the sheer diversity of serotypes that we know exists. So here we're dealing with Illumina HiSeq 2500 data of 151 base pairs paired end breeds. So we can tabulate this. So we have our breed, our foot and mouth disease treatment. So here you can see for each of the groups, you've got 3 foot and mouth infected and 2 uninfected. So that's a total of 9 versus 6. And that comparison, when you consider the breed, the treatment, and then the within sample error. So therefore, we're considering that our level of expression of either a transcriptor gene is proportional to some function of the breed, the treatment, and the error, the sampling error, and other errors that are inbuilt into the variation observed for each particular sample. You can see in terms of samples, there's a large number of reads produced for each, and when we do QC on these reads, we'll find that the quality is pretty good. However, what we will do is consider how we can improve this work. So their filtration steps are unclear, basically, in the paper, and that QC is important. They use Top Hat, which was last updated 7 years ago. It's not a recommended tool anymore. They also use DSeq2, which had insufficient detail on precisely how they tested for differentiated expression, and we also DSeq2 is prone to very high rates of false positives. And that's especially accentuated when they had a full change threshold of just 1.5 instead of 2, which is more typical. That again will increase the rate of false positives. We also it's also obviously evident here that we have very small sample sizes, and the testing approach to combine the breed effect as well as the treatment effect, and their interaction also is not really considered in detail here. And finally, they try to validate the RNA-seq with RT-PCR, which is kind of inverted thinking, because RT-qC-PCR is less not as sensitive as RNA-seq. And we can also see this evident in their volcano plot, which is quite skewed when they're looking for changes in expression, and it kind of tells you that something is amiss. So what we will do in our lab is we'll do proper QC. We'll use proper research valid tools. We'll use appropriate ones that are relevant for this analysis. We'll also be aware that not everything has to be labeled as significant. We'll also consider the effects of the sample sizes and how that affects this kind of piece of work. We won't do RT-qPCR because it's not valuable for this kind of approach. And we'll also look at volcano plots as well. So what the suggestion is for you is that maybe we'll just look at 5 samples each, just 5 samples for one breed. You might use the Malagida ones, for example. So you're comparing 3 versus 2, just for example. And the reason for this is that these samples, when you transfer them over a table like this, it just takes time to go through. So we want to have time to finish the lab. So doing 15 may take too long. What we will do is use the Callisto Salute pipeline as well as Lima as well. And there we're looking at quantifying with Callisto and that is based on the idea of pseudo alignment, as mentioned earlier, and that in essence is looking across the different de Bruijn graph for k-mers pseudo alignments, so that we have this de Bruijn graph here where each node is a different k-mer, and we have a chain of them like this. So we might find that we have certain ones that belong to the same class, and therefore we don't need to align our read at these three here that belong to the same class. We can align at say this one, because it has two nodes extending from it, and therefore we'll need to map our reads to this one and this one, because these are different paths. So you don't have to exhaustively align to every possible k-mer. What we can do then instead is bootstrap the efficacy of this initial quantification approach repeatedly to get an estimate of our uncertainty associated with that. That's visualized more effectively here, where we have three different transcripts here in pink, blue, and green. So black is our read, and these are represented in a de Bruijn graph here in B, where we have different k-mers represented by the nodes, and the edges join them. And you see this split here, where you have two different transcripts that are produced. You see for green here that they're missing this just chunk here. So we have two different paths, and we're checking if the reads of the black part map to here. But for the black part, we don't actually need to check all of them. We can skip some, and that's shown by the dashed lines there. And we can check vectors 1, 4, and 5 only is sufficient to test for evidence of the blue-pink isoform pair, and contrasting that with the green option. If we have a path going down for vector 1 and this one, we'll know that's the green path that we're traversing. So that efficiency means that we can just classify reads based on the k-mers that they match. What we'll eventually get from this process is a table like this, where we have the transcripts, their lengths, which we can see there, their effective length, the counts, and then the TPM in this final column, which is our main part of interest. So that's our abundance.tsc file, and we get the transcripts per kb per million. We'll also generate a run info JSON file, which just contains the setup for our study. We'll also get abundance.h5 files, which contain the bootstraps, and we'll look at them further then. And we'll do that using sleuth, and that sleuth models different sources of variance to predict which transcripts and genes are differentially expressed. So here we have biological and other factors that can affect our model. As mentioned earlier, we've got read and the treatment, as well as errors, different sources of error inside here. And we have this negative binomial model with two key parameters here. And then we have to consider our raw variance that we get, and then the smoothed biological variance, as well as the inferential variance as well. So the biological variance runs us from the actual changes between RNA content and the technical error, and the inferential variance rises from our analysis. So if we have between sample raw variance, that results in a small estimate, results in a small estimated biological variance. We have to consider how this affects the uncertainty in quantifying the different samples. So you kind of have two different models that we can compare: a null model where the changes in expression are zero, in effect, an alternate model where there's actually big changes. So with sleuth, what we're doing is using this R-based tool to compare these two different models. We're linking that data using a table like the one I showed you before to link it to the Callisto data. We're looking at the bootstrapping data at the abundance.h5 files. We'll create a folder for that data, like the table. And what we'll get at the end, we'll try to get is a plots like this, where we can look at particular transcripts in this case. And we're looking at the relative EST counts. And we can look at the different values that we have here for the FMD infected and see if they're different from the uninfected ones down here. So you can see here for each of the breeds, they're in trios and then in pairs here, is that we see that the expression of the FMD infected is higher than the uninfected in this particular example. And what we'll be able to do then is use the likes of dplyr to look at transcripts where you can see that the Q values and adjusted P values are below the thresholds that might be of interest to us. And then we can also compare between different breeds. So this is another example of a transcript where we partition between the MG breed and the other ones. We see that on average, they tend to be different, bearing in mind that we just have a 5 versus 10 comparison here. And we also compare then for the same transcripts, which you can see up here, what are the effects across FMD infection here, across all of them, where we kind of see there's a bit of a difference there. Compared to that within breeds, you see they got 2 versus 3 here, 2 versus 3 in Holstein and Friesian, and 2 versus 3 in the Malna Gita ones. So that kind of completes the key aspects of our lab for this Callisto sleuth approach. In the next video, we'll talk about the Lima approach.